Hello, everybody. Today I'm going to tell you the story about how the evolutionary loss of a single glycan shaped host-microbe interactions. This story, or most of the story, was recently published in January and only last week was featured as the cover of this month's issue of Cell, Host, and Microbe. Glycans are sugar molecules Glycans are sugar molecules that are linked to either proteins or lipids. The alpha-1,3 galactosyltransferase, or the GGTA1 enzyme, synthesizes alpha-gal by this reaction, which consists of two galactose rings linked by an alpha-1,3 bond and is depicted as such. Now, the evolutionary significance of this glycan lies that during primate evolutions, ancestral primates expressed functional GGTA1 and thus expressed alpha-gal. However, after the split from New World monkeys, Old World primate lineage acquired three different loss-of-function mutations, rendering the gene non-functional, such that the Old World primates did not express alpha-gal. Now here we ask two questions. First, what was the advantage of loss of alpha-gal expression? And second, why was it unique to primate evolution? We hypothesize that loss of alpha-gal alters host-microbe interactions. So for this, we used a mouse model in which the mouse carries a deletion for GGTA1, which mimics the human loss of GGTA1. As a model of polymicrobial sepsis, we used cecal ligation and puncture, whereby the cecum is perforated, allowing the contents to disseminate and induce polymicrobial sepsis. We then assess the survival and bacterial load in the organs. Upon CLP, you can see that the GGTA1 knockout mice in black have a tremendous survival advantage. And this was associated with a up to 10 to the 6-fold decrease in bacterial load in all the organs, suggesting that loss of GGTA1 confers protection against polymicrobial sepsis via a mechanism of resistance. Now, since the outcome of CLP is influenced by the microbiota composition, we next sequenced the fecal microbiota of GGTA1 knockout versus wild-type mice. The principal component analysis for both unweighted and weighted UniFract showed tremendous differences in the bacterial communities of wild-type versus knockout mice, as you can see here. The LEFC analysis showed several taxa enriched in the wild-type microbiota here in green and versus the knockout microbiota here in red, which included several pathobionts, including members of the Helicobacteriaceae family. We then asked whether loss of GGTA1 actively shapes the microbiota via an antibody-dependent mechanism. For this, under controlled conditions, we crossed wild-type females with GGTA1 knockout, JHT knockout males that lack mature B cells, hence cannot produce antibodies. The F1 double heterozygous litter pups were then crossed to generate F2 litter mates, which were either wild types with and without antibodies and knockouts with and without antibodies that acquired the same microbiota from the wild type mothers. We then crossed them to generate the F3 progeny, whose microbiota we then sequenced. Principal component analysis for weighted UniFract shows that the wild-type mice in the presence of antibodies in blue differ significantly from the knockout mice in the presence of antibodies in red, showing that indeed loss of GGTA1 shapes the microbiota. In the absence of antibodies in brown and in green, this difference disappears. The weighted UniFract distance between the knockout mice with and without antibodies was higher than the wild-type mice with and without antibodies, showing that the antibody-mediated shaping of the microbiota is enhanced in knockout mice versus the wild-type mice. Now, within the knockout mice, in the absence of antibodies here, you can again see an enrichment of the Helicobacteriaceae family, which contains several pathobionts. We then asked whether loss of GGTA1 confers resistance to sepsis via an IgG-dependent mechanism. For this, we took GGTA1 knockout mice that lack adaptive immunity. We then transferred IgG purified from the serum of wild-type versus knockout mice. We then challenged these mice with a cecal slurry containing microbiota that has not been shaped by antibodies, and then we looked at the survival. The survival curve shows that the group of RAG2 double knockout mice that received vehicle survived as much as equal to the group that received wild-type IgG. However, the group that received to the same extent IgG from knockout mice survived to a much better extent, suggesting that the IgG from wild-type versus knockout mice are functionally different. These IgGs were predominantly of the IgG2b subclass, and they were produced to the same extent in both genotypes. Now, IgGs are glycoproteins. It is known that in wild-type mice, the Fc portion of the heavy chain contains glycan structures which expresses alpha-gal at the terminal end. We therefore asked whether knockout mice that lack alpha-gal expression on the Fc structure. This turned out to be indeed true, as shown by the Western blot analysis here, where we see decreased signal in the heavy chain of the knockout mice. Now, therefore, we asked whether loss of alpha-gal from the Fc-IgG glycan structure modulates IgG effector function. Now, since these IgGs were IgG2b, we then asked whether it influences binding to its cognate receptor, Fc gamma receptor 4. Surface plasmon resonance analysis showed that indeed the binding affinity to Fc gamma receptor 4 is enhanced in knockout IgG versus wild-type IgG. Now, as I said before, loss of GGTA1 is a unique event in primate evolution. This suggested that it was probably associated with a fitness cost. For this, we analyzed literally hundreds of breeding pairs of wild-type versus knockout mice and assessed their reproductive output over a period of 5 years. It turns out that knockout mice produce 66% less pups during their reproductive lifespan, and this was due to an earlier onset of reproductive senescence. However, since loss of GGTA1 was in fact selected and fixed in old world primate evolution, we predicted that the survival advantage against infection outweighed the reproductive fitness cost. Mathematical modeling showed that in the absence of infection, there is indeed a fitness cost associated with loss of GGTA1. However, this cost is outweighed in the presence of infection. And such, in the presence of high risk of infection with a highly virulent pathogen, the protection conferred by loss of GGTA1 moves the species beyond this black line into a parameter space where these loss of function mutations are selected and eventually fixed. In conclusion, loss of GGTA1 shapes the microbiota composition by antibodies. It confers resistance against infection by IgG2b. It enhances IgG2b binding to Fc gamma receptor 4 and effector functions. And then it precipitates reproductive senescence, which is outweighed by survival advantage against infection. In the end, I would like to thank my PhD supervisor, Miguel Soares, without whom this work would not have been possible. I would also like to thank the members of the Inflammation Lab, our collaborators, the IGC core facilities, and the incredible IGC community. Thank you.
Thank you very much. Ask the audience if you have any questions. So maybe I can start. So do I get it right from the very quick mention of your next publication that the loss of the IgV might also lead to a divergence of the microbiota, gut microbiota?
Yes. This paper is under review right now, but we have shown that loss of GGTA1. I've shown in the slides that loss of GGTA1 shapes the microbiota via antibodies. And in the absence of antibodies, several pathobionts like Helicobacter species become enriched in the gut. And we believe that this exerted a major selective pressure in selection of these loss of function mutations in old world primates.
Mm-hmm. We still have a bit more time if there are questions from the audience. Yes, so maybe one follow-up question. So do you think that the difference in the microbiota is because of different composition of resources in the gut, or because of different functioning of the immune system?
So our work has been based on antibodies. So I've shown that in the absence of antibodies in a controlled environment, knocking out antibodies shapes the microbiota in knockout mice more than it does in wild-type mice. But alpha-gal is a ubiquitous glycan that is expressed on the surface of all cells throughout the body. So of course, alpha-gal is also present in the gut epithelium, also in the mucus of the gut. And it is in direct contact with members of the microbiota. So it's not very hard to envision that losing alpha-gal, a major glycan from the mucus and from the gut epithelium, would shape and alter microbiota via an antibody-independent mechanism as well.
Mm-hmm. Good. Thank you very much.
Thank you.